This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 5. Hello, everyone. I'm Vincent Racaniello, and I'm with Dickson Despommier.
Hello, Vince.
How's it going, Dick?
It's going very well, thank you.
I'm glad to see you again in a parasite mode.
Yeah.
Some days we're in a virus mode.
That's right.
And some days we're in a parasite, which is, I finally have understood your idea of broadening the virus
Groovy.
to include parasites. To include parasites.
As well we should.
And the parasites are fascinating, but
Yes.
that's because viruses are a subset of them.
Right.
So last time we talked about trichinella.
We
I
did.
think we've done 2 shows on trichinella now.
This is true.
The first was a general. The second
Was the life cycle.
life cycle, which was terrific.
Glad you liked it.
Is trichinella an eradicable disease?
You could eradicate it from the domestic scene, but you could never eliminate it from the wildlife. Because everything lives and then everything dies. And when it dies, it either decays and disappears into the ground and gets recycled, or the meat of the animal is consumed by scavengers.
Mm-hmm.
Mostly it's a scavenging world out there. Animals don't have to be able to hunt and kill to make a good living. All they have to do is wait for something to die.
So you'd have to eradicate all the animals, basically.
I'm sorry to say that, but that is true. But we're capable of it. It would be wrong to do that, however. That was a joke.
I'm not sure we could find every last one, though, Dick. Dick, is that synonymous with pork tapeworm?
No.
No, what's pork tapeworm? See right here on Wikipedia, pork tapeworm.
Pork tapeworm has got a
Totally
different phylum.
Right, because I just had been looking up eradication
Right.
of infectious diseases, and only one has been eradicated.
And that is smallpox.
And polio and dracunculiasis. Dracunculiasis.
That's right.
is ongoing.
It's got a potential.
That's a parasite, right?
It is. It's a big worm.
We're going to talk about that at some point.
We will.
And then they say here others are lymphatic filariasis,
Yes.
and pork tapeworm.
Correct.
And
that are potentially eradicable.
But pork tapeworm is not the same as trichinella, right?
Oh, it's as far from trichinella as rubella is from spirochetes.
Okay.
So there.
So we'll get back to it, right?
We will,
Okay.
if you'd like.
We want to cover everything.
We do.
All parasites.
We have the rest of our lives to do this.
Yeah, if you drop dead, I have an issue. And if I drop dead...
You can just sit here and read this book if you'd like, but that would be wrong also.
Well, if you left us, which we don't want to happen...
We don't... No, I'm not planning on going.
It would be a problem for TWiP.
Well, that's very nice of you to say that, actually. I think my wife might feel a little bit of remorse
and
also.
TWiP would be difficult.
That's right. And sorrow.
But let's not be morbid.
Of course not.
And let's proceed with trichinella.
Right.
This will be the third and last episode on trichinella?
Not necessarily. It depends on who wants to know more. But if you go to the website called trichinella.org, we will continue this discussion.
What is this trichinella 1835 to 2007?
Well, the 7 should be changed to a 9.
That's just the amount of knowledge you've got stored here.
That is
correct?
exactly right.
It's not when it existed, because it was in existence before 1835.
That's right.
presumably.
This is a reference to the world's literature, which we've posted most of on this website.
Okay.
It was discovered in 1835, and I think we've gone through that part of this.
Right.
And it was a fascinating discovery, of course. And then we've gone through the life cycle as to how it evolved into a parasite that could live for as long as, let's say, 20 to 30 years inside of the same host without being destroyed. And we were discussing, as I recall last, the relationship between the development of the nurse cell, which is the house that this parasite builds from scratch out of a portion of striated skeletal muscle cell, So this worm is a master builder, but the worm itself does not secrete any portion of its house. It reprograms the host's genome to make all of the elements for its house.
So it must secrete something to do that, right?
It definitely does. So I would like to today. Let's try that.
Try that again.
I would like today to discuss the row of cells which lies in the front part of the worm, both in the larva and in the adult, called the Stickosome.
All right. Can we see a picture of this?
We can. It's on the website, actually. If
we're at trichinella.org.
We will.
And where shall I go?
We should go to the biology section, which is the first section on
biology.
the top. Yeah, there you go. Go to the third image and click on that.
It's too small for my old eyes. Oh, now it's big.
I don't think so, Vince.
Dick,
I think I can
did you draw this?
Well, no, I actually didn't, but we colorized it.
We should say we are at trichinella.org, and we clicked on biology.
Right.
And then there's a diagram labeled infective muscle larva.
Correct.
And we clicked on that, and now it fills the screen, and it's multicolor.
And not only that, if you scroll down through the legend, you will see that portions of the worm correspond to each one of those colored views.
So muscle cells are brown.
Yes.
And hindgut is light blue. You have 3 shades of blue. We should point out 4 shades.
We do. We have to, because there are so many different structures.
Did you draw this?
I did not.
Someone else did.
That's right.
Have you plagiarized this?
No. We give credit. We always give credit. The people who drew this, though, had no concept of what the row of cells called the Stickosome did.
And where would that be in this picture?
Well, you can start in the mid part of the worm.
It's about right here.
Yeah, that's right.
Green. There are a couple of green cells which are labeled
alpha 0.
No, these are the alpha zeros. Here, the light green.
Yeah. Well, what are those called then? Those are called the
gamma.
There is a color for those, no?
No, it's not in the legend.
Oh, that's too bad. We'll have to correct that then.
It's not gamma cells.
Yes.
These are gamma cells?
Those are gamma cells. They are labeled. Come on, Vince.
All
They're
right.
there.
Well, the colors are a bit off.
It's a dark green.
Dark green, they're gamma, and then above gamma is red.
Yes, and the red corresponds to?
Stickle cells?
No, alpha 1s.
Now, what does alpha 1 refer to?
Well, we'll get to that in a moment. And then the blue cells.
Dark blue here?
Yeah.
Beta.
That's correct. And then the light green cells.
That's alpha 0.
Correct. And the purple cells.
Alpha 2. This sounds like a science fiction thing here.
It does, doesn't it? Well, for lack of a better term, because if you're asking me what each one of these cell types does, I can in general tell you what it does, but in specific, I cannot tell you what it does.
What's this light blue thing going the length of the worm?
That is the worm's gut tract.
Which is the head? The
head is the thin part.
Right here?
Yeah. And that little red thing that's sort of diagonal there, just a little ways down, that's its nerve ring. That's the brain of the worm.
I was just going to say, where's its brain?
It's right there.
It's about my size. All right, so this is the digestive tract, which goes from the head of the worm
all the way to the end.
All the way to the end. It gets bigger after the gamma cells.
It does. It expands out into a midgut.
And we have a lot of genital primordium.
We do.
Because I guess reproduction is key.
They eat and sleep and reproduce. That's it. That is their life.
I eat, sleep, and study viruses. I guess humans have a problem, right?
But they've sort of diverted away from the original life cycles.
So these are what you see in a microscope when you cut open a worm.
That's right. Or even look at it through Nomarski optics, phase interference.
And how do we give them different labels? What's the criteria for that? Morphologically?
Well, yeah, they're all based on morphology. And notice the title of the drawing is the morphology of infective muscle larvae.
So this is the stage that's in the muscle.
That's correct.
It's in a nurse cell in this?
It is. It is. So if you go back, you can actually see the nurse cell. No.
Biology?
Yep. Then you have to go to under
life cycle.
The nurse cell. No, it says the nurse cell.
Ah, sorry, I didn't see it.
That's quite all right, Vince.
Okay.
And there it is.
I didn't know that the nerf.
That's a Nerf ball.
A nurse cell was surrounded by small blood vessels.
It is, and we're going to get to that.
In all the photos we've seen, it's just been this spiral structure.
Correct. That is correct.
But now I see something new.
Right.
Venules.
We are placing the worm in context.
Mm-hmm.
So the nurse cell actually develops from a portion of muscle cell as shown in the red diagrams below that.
Schematic of a larva invading a muscle cell.
That's correct.
Which we talked about last time. It invades the muscle cell and sets up this nurse cell.
That's it. Now, all that blue clickable stuff on the left.
You can look at photos.
You can look at photos.
The actual real stuff as opposed to a diagram.
Correct. To show you that we're not just making all this up.
I think people should know that.
Well, I know they should know that, but this has been observed very few times, by the way.
The actual invasion of the muscle cell?
I will tell you that when we first saw that, we went out and bought a bottle of very fine wine.
Did you take these photographs?
I did, yes, I did.
So it's a matter of timing, right? This isn't a mouse?
It's in a mouse. The way to get around the timing aspect is to isolate the newborn larvae all at once and then inject them into a muscle bundle. And then shortly afterwards, preserve that muscle bundle at the site of injection and start looking for these events.
Yeah. And you hope you get it, yeah.
You do hope you get it.
No, that's the way it works sometimes. So that was...
that was what?
Oops.
How many minutes?
That was worm entering a muscle cell.
So that's time 0.
Which is step 2.
Right. Step 3.
And step 3 is click here. Oops, sorry.
Right.
Larva 10 minutes after penetrating muscle.
Notice now the edge of the muscle cell has been completely exploded by the entry event, and the worm is now firmly embedded in the skeletal muscle elements. So as opposed to viruses, because I think a lot of our listeners will know that we also participate in another podcast called This Week in Virology. In Virology 101, we've covered the entry process of viruses, and they're a lot less violent than the entry process of this parasite. There's an attachment phase, and then there's an invagination of the membrane, and then all that stuff occurs. This parasite just bowls its way into it.
This is a multicellular event here. Right?
Oh, yeah.
These are many, many cells involved. So it has to break down the cellular structure, the tissue structure.
Depends on how you define a muscle cell. A skeletal muscle cell is a very long, multinucleated cell.
Right.
It's partitioned off very much like a submarine. All right? So this worm enters a region of that very long cell and starts to influence the cytoplasm on both sides of itself by secreting something into the milieu. And that causes the rearrangement of the muscle cell in that zone to begin the process of nerve cell formation.
Of course, you don't feel any of this, right?
These worms at this stage, when they're migrating, don't always migrate into the muscle cell. They can migrate into your brain, into your eyes, into your pancreas, everywhere. And in fact, if you have a heavy infection, you definitely will feel it. What happens is that the worm exits from the capillaries and you get local bleeding events and the tissues begin to swell up. So you get edema. And it's particularly noticeable around the eyes. So it's bilateral periorbital edema. It's one of the signs that the doctors will look for here. Okay, so back to the life cycle here. Because knowing how this worm behaves is to know all about the stickocite cells. Okay?
Which again are
this row of cells that contain 5 different kinds of cells.
Which we pointed out previously.
We did.
The different colors, right?
That's correct. Now, each color represents a different variety of secretory granule.
Okay.
So if you could imagine, for instance, those that are familiar with mammalian physiology and biochemistry, the pancreas. The pancreas of a mammal contains islet cells, which produce insulin, and zymogen granule cells, which contain all of the digestive enzymes that you need to process your food.
Right.
So if you look at the pancreas, it's an endocrine gland and an exocrine gland. It's double. The exocrine gland aspect means that all of these zymogen granules, when we eat a meal, as by the way we just did, didn't we?
It did.
So our zymogen granules are going crazy right now, exiting the assigner cells, which they're called. And the products located within each of these granules then fuses with the duct and dumps the proteins out into the intestinal tract, where they contain DNAases and RNAases and cathepsins, and amylases. All of these protein enzymes are to help us reduce our food down into the elements that we need to build them back up into our tissues, right? Okay, now this worm is no different. This worm has similar needs. It has to grow from a small larva to a very large larva extremely fast, because we covered that in the life cycle. In fact, it can actually achieve infectivity before it achieves maximum growth. So we would call that precocious.
Achieves infectivity, you
Yes.
said?
So
What does that mean?
I'll tell you what that means. It means that, let's do the experiment and I'll explain it. We take gravid female worms out from an intestine, from an infected mouse.
Gravid?
Meaning they've got lots of larvae inside their
Okay.
uterus. And we leave them overnight in nutrient medium,
Mhm.
like medium 199 and calf serum. And they give birth to their larvae.
Mhm.
Then we can separate the adults from the larvae because there's a big size difference. Then we can centrifuge the larvae down into a small pellet. And now we can get literally millions of newborn larvae. We can then take an aliquot of those newborns, and with a tuberculin syringe, I can inject the thigh muscle, the hind end thigh muscle of a mouse.
And bypass all the preceding steps.
Correct. What I want to do
right
is
into the
initiate
muscle.
a synchronous muscle infection.
Meaning you want many cells infected at once.
And all at the same time.
Okay.
Whereas in a natural infection, it's infected over a 9 or 10 day period.
It's stochastic, right?
Right. So I'll never know
Random.
how old that is unless I have a size measurement of the larva, because I know exactly how big the larva is at which age.
We do similar things in virology.
Of course we do. When I say we, I mean the scientific fraternity.
Yeah, sure.
So if you think of this as a very large virus, then the kind of research that you and I have been carrying out for the last 20 years is very similar. Only we're asking less, I think, molecular questions, because our parasites are so confounded by their multicellularity that we now are dealing with a multicellular organism living within another multicellular organism. It's not like a simple virus, by comparison, of course, where the genetic material is being replicated and then you get another virus particle. This parasite does not reproduce the same way, but what it needs to do is all of the things that a virus needs to do. It needs to survive long enough to go.
The problem here is the host, because C. elegans, which is another worm,
it's free living
worm, very different.
It's not a parasite.
That has been studied
ad nauseam.
extensively.
That's right.
At using similar molecular approaches as
correct.
viruses. But since we don't have a host to worry about, it's easy.
Of
and
course.
the problem with trichinella is that you have to grow it in a host to really understand what's going on.
And remember, we already said early on that the trichinella contains actually more genes than Sinusoids, because it needs to be able to do more things than a free-living animal.
Okay.
All right. So part of the life cycle of this parasite involves long-term survival. And the strategy that this parasite employs to do that requires that the host surround its home that it builds, this nurse cell, with a vibrant, active circulation.
Why does it need circulation?
We'll get back to that.
Good. Shit, I didn't know that.
You're right. So this is a very unusual deal with parasites. The parasite actually controls the angiogenic process in the host.
What does angiogenic mean?
It's a term applied to the formation of blood vessels. So when you cut your finger, for instance, and you get a loss of oxygen transport on both sides of the cut, it creates an event called hypoxia. Hypoxia is enough to induce the cascade of events that leads to new vessels forming to connect back up again to relieve the hypoxia.
So it's part of the healing process.
It is the healing process. It is exactly right. So studying the healing process has taught us how tissues repair themselves. But this is a damaged tissue. There's no question about it. The parasite goes into a muscle cell and damages it, but it doesn't kill it.
Doesn't want to kill it,
It doesn't
of course.
want to kill it. What it does is it wants to hijack it. It wants to take a little piece of your host and keep it for itself. So that's the secret to this parasite. You have to think like this parasite in terms of they have 14 days to do this. That's all. Within 14 days, they have to be able to they have to be able to go to the next stage. If they can't by that time, then the parasite dies. But 14 days is not a long time.
Depends.
Even in a host that has received a lethal infection, they don't die in 14 days. they die in 25 to 35 days.
Mm-hmm.
So this still gives plenty of time for some of those worms to grow up enough so when that host dies, it'll be consumed by other animals and therefore transmit the infection on.
Right.
So the survivability of this parasite is incredible. And that's why it's found everywhere. This parasite covers the world.
This is the most successful.
One of
life
the
form.
most.
One of the most.
Yeah, the other one is also transmitted by the eating of meat, and it's called toxoplasmosis.
Another very successful life form.
It's an extremely successful, and we'll discuss that too. Okay, so let's go back to the diagram of the stickosite cells. Right. Now you're going to ask me why we have different colors for different cells.
Yes, I'd like to know that. Because as a virologist,
Sure.
I would say you had to stain them with antibodies to antigens to make them different colors. Or
You'd say that.
they had different origins from
That's
the
right.
different somehow distinguish them. So what
That's
did you
correct.
do here to make them different colors?
Well, what we did was we did something very clever. Actually, we did something very stupid to begin with, and namely that was what I did.
You asked them.
What I tried to do was to take sections of the worm at different levels of the stickosite,
Mm-hmm.
or stickosome, I should say, stickosome, which is a Greek term for a row of cells. That's what that actually translates out to. And I tried to describe which cell types I was seeing in various sections. Sometimes I'd get an exact cross section, but a lot of times I would get oblique sections, okay? And it was difficult to put all this together into a puzzle so that you could make a complete picture out of this. So although I was the first one to publish on the morphology...
Oh, so this is your doing?
Yeah. On the morphology...
I'm humbled and honored to be in your presence.
Stop it. No, because what I'm about to tell you trumps this beyond belief. And so imagine trying to get electron micrograph pictures of this entire length of the worm. Now, you mentioned Sinusoids elegans. You can buy an atlas
Mm-hmm.
where they've got the head of the worm here and the tail of the worm here, and each page is a section of worm.
Do you have that for Trichinella?
We do not.
Do it.
It's already been done, and I'll tell you how they did it. Instead of publishing it page by page by page for each section of worm, there was a Japanese worker by the name, and still is, by the name of Yuzo Takahashi. He's a good friend of mine.
Mm-hmm.
What he discovered was that as this worm, when you isolate it from its nurse cell by digesting it away using pepsin and hydrochloric acid, which is the conditions in the stomach by which this parasite achieves its life cycle, it will coil and uncoil. Now, if you were to fix it in a coiled position, it's like taking your thumb and forefinger and making the OK sign with it.
Got it.
Then if you lay it on its side and let it rest on a glass slide, and then put a cover slip on top of that, and then begin the process of embedding for electron microscopy, what you will end up with is a glass slide with a cover slip on top, and the worm is in the middle, completely embedded in EPON, ready for sectioning.
EPON being some resin that
It's a plastic resin. That's
you
right.
embed things in for EM.
That's correct. So what he did was he popped off the top of the glass slide, and he now had a perfect worm that he could get a longitudinal sagittal section on. He got the whole worm in one section.
Sagittal meaning the length.
That's right.
Of
Right
the
through
worm.
the middle. If you were to make a round circle with your fingers and cut that directly in half.
As opposed to a transverse, which would
That's
be
correct.
cutting it. in
Me,
bits.
I was doing cross sections, and he did a total longitudinal section of this worm and described every single cell type. I missed 2 of those cell types.
So he was in general agreement with your...
Oh, yeah.
So you name them basically morphologically, right?
That's right.
Beta and alpha and
That's
alpha
correct.
0. And
That's
he pretty much agreed with a few differences.
He described 2 new cell types.
So the picture we're looking at here is his and yours
No,
together?
it's an older drawing made by a guy by the name of Wu and Villela, and we colorized their drawing because it was such an accurate drawing. Now, if you go to the website homepage, here's a drawing. Oops, you won't. Yeah, that drawing was made in 1860. What's the date?
You'll have to zoom in
Let's zoom in.
because it's too small for our old eyes.
It is. Well, I can almost tell you when this drawing was made. It was made in the early 1860s.
Say 1800s because we can't read it. Yeah.
No, it was made by Virchow. And the same row of cells can be seen here
Yeah.
as shown by Villela.
So he drew this by hand.
He drew it by hand, and he was a wonderful observer of detail. Okay, so now we can go to the electron microscopy of this cell type, and we can look at it, and I can show you some pictures of what these cells look like under the electron microscope.
Okay.
And
Where
for
should
that,
we
we
go?
have a gallery.
Gallery of images.
That's the one.
So we're still at trichinella.org.
We are.
We clicked on gallery, and there's a funny picture of Dickson and Yuzo,
That's right.
who we've just talked about.
Cartoons. That's right. So he's contributed 165 images, and I have 444 images.
All right, so where should we go next?
So you click on my pictures first, of course. Now, which one do you want to see?
Whole worms?
Let's look at EMs. Aha. Lots of choices. So the mature larva, let's look at the stickosites. And there they are. So now you can just review these electron micrographs.
So these are electron micrographs.
And that's a schematic that you pulled up that was drawn by Robert
cross-section.
Demarest.
Mm-hmm.
Yep. And it's got all the parts labeled and everything. So now if you click on each one of these other pictures, of course,
We get an EM.
you get a high resolution EM, and you can actually scale up. I think you can zoom in and zoom out on all these to let you look at some of the details. Yep, you can go even higher than that, sure. And I've put these in under high resolution conditions so that you can actually see a lot of these details.
So let's see. I've clicked on the second EM here.
That's right. Now those are alphas. Those are
alpha
alpha
cells?
cells.
There's a cross section, of course.
Yes.
Okay.
You notice in the alpha cells that if you didn't know what you were looking at and you thought maybe you'd seen these before, you would be right in that if you looked at eosinophils, they have a granule that's very similar to this. It's not doesn't contain the same things, but has a similar morphology. You'll notice also that some of these have very, very repetitive and crystalline-like structures.
Mm-hmm. These are alpha cells.
Yes, these are alpha granules in the alpha stickosite cells. It's reminiscent of viruses.
Yes, the arrays of viruses being made
That's
in
right.
cells.
What you have here is a single protein
Mm-hmm.
that's crystallized inside of a larger granule that has another protein that's another crystal inside of that.
So do we know the composition of these cells? These alpha cells.
Vince, let me tell you what I did at Rockefeller,
Uh-huh.
because this relates to my postdoctoral experience. When I first came to Rockefeller, which was in 1967, I had a lot of these electron micrographs with me. And one day, I worked in Dr. Traeger's laboratory.
Bill Traeger.
Yep, William Traeger. He was famous for malaria.
Yes.
That's right. But in his laboratory also was Dr. Philip D'Alessandro and Dr. George Jackson. And I actually was working with Dr. George Jackson, but Bill Traeger was in charge of the whole group. And I had my electron micrographs that I had just taken last year during my summer between the time I got my doctoral degree at Notre Dame and the time I was to start at Rockefeller. I took a lot of EM pictures. I had the whole summer to do nothing more than just do something that I wanted to do. And I wanted to know more about this row of cells because I found them intriguing. I had spread them all out and I had categorized them according to the ones that I could tell. These are the same, these are the same, not knowing what they did. And in the lab comes a guy by the name of Miklos Mueller. Now, Miklos and I became very fast and solid friends. He worked in Dr. Christian Dedoux's lab. Some of the listeners may know him, Dr. Dedoux, for his famous work on lysosomes and peroxisomes, for which he received a Nobel Prize. So Miklos, not having met me before, introduces himself, and he looks at these pictures, and he said, these are very interesting pictures. What are they of? And I said, well, these are electron micrographs that I've taken of a worm that I did my thesis work on called Trichinella. And Miklos knew vaguely what Trichinella was, but he wasn't very, very sure of this because Miklos is a protozoologist by training from Hungary who worked in the Carlsberg labs and worked on Trichomonas. So he's very famous for his work on trichomoniasis. But he loves to work on granules of various kinds, all right? That was his specialty. His specialty was intracellular granules. And, of course, Trichomonas has one of the most famous ones now called the hydrogenosome, which, if they could clone and sequence all of the enzymes necessary to make molecular hydrogen and put that into bacteria, we would have fuel for the rest of the world, because they could make as much molecular hydrogen as you could ever use by microbial process. So that hasn't been done yet, but I'm suggesting that should be done. All right, so Miklos takes a look at my pictures and says, oh, they're from a worm. And I said, yeah. He says, oh, this is a very interesting worm. And then he said, what do those granules do? And I said, I don't have a clue. He said, would you like to find out? I'm not sure. It's exactly what he said. Would you like to find out? And I said, yes, I would love to find out. He said, well, why don't you come over to our laboratory and talk with us about what you're doing, and we'll see where that might lead. This was the first month that I arrived at Rockefeller. Vince, that was the turning point in my life, because I did do that, of course. At Rockefeller, you have maximum freedom to do whatever you want. It's a great place for that, and especially as a postdoc, because you don't have any obligations. So I went over and I met Christian de Duve, who's a wonderful, gentle, intelligent, caring, funny. He's an ideal leader for a laboratory, and so is Bill Traeger, by the way. They were both very generous people with their times and energy, and listened to what you had to say. They didn't just didn't wait for you to stop talking. They were actually interested in what you had to say. And I brought some pictures with me the third time I went over there. I was with Miklos first, and he showed me what he was doing, and then he said, but I have some time that I can devote to some other projects, and I like this project because it looks like these granules would be very easy to isolate and then to study as to what they might be doing for this parasite. So I said, well, how would you do that? Not knowing much about cell biology at that point. He says, oh, we can handle this. We've got lots of very nice techniques for this. We use sucrose gradients, and we use continuous flow rotors. And he started to talk about things that I knew nothing about, and I was fascinated by the possibility. But the other thing that I was really fascinated by was the fact that they didn't stop at knowing what the granule did. They wanted to integrate that into the biology of this unit. Of
course.
And so they did the morphology of this as well. They were very interested in structure-function relationships. So they had the finest imaging equipment of the time, and in that case, it turned out to be a Philips 300 electron microscope, which had just come out. And because he was at the forefront of his field, he bought one.
Sure.
So he said, and we'll let you drive this thing too. You can drive. Let's
nice.
Like getting behind a wheel of a Ferrari when you've been driving a Model T Ford all your life. And I just looked at this machine in this special room that had speakers for music and everything, and you were all by yourself in there, and you were exploring like the surface of Mars, it felt like. I had so much fun at that part of this.
Looking at these worms.
Yeah. So we outlined a program of research which was based on isolating as many of these larvae as we could get our hands on, and then doing cell-free extracts of these larvae, and then isolating each of these fractions of particles based on their isopycnic densities in sucrose. And to make a long story short, we ended up with a series of research papers, including ones which broke open each granule type and then analyzed for the presence or absence of antigens based on an immune response that a mouse might give to the entire infection. And sure enough, we found each granule type contained a separate set of antigens. They weren't the same. They were totally different. And these are proteins. So when I say antigen, I don't mean they were antigens in the sense that that's all they did. They were not just antigenic.
Sure.
They were functional molecules for the life cycle of this worm, which were anagenically distinct.
Right.
So it's like looking at, I know that I've heard some of the virologists talk about early, middle, and late proteins during a viral replication cycle. And this is E2A and E2B, and what does that mean? Well, we don't know, but this protein comes up first, and then this one comes up second. And, it's the same with this parasite. We've got a lot of knowledge about where the granules are made, what the cell types look like, and something about the protein contents of each of the granule types. Although with modern proteomics, if we had had that then, we would have described all Well, I'm guessing now that there are 3 to 500 different proteins at this
So
parasite.
you're telling me that you discovered the proteins, where they were located in specific cells,
Yes.
and we don't know what they do to this day.
Well, we don't know what most of them do.
Most of them, some maybe, but not all.
Correct. This
And
is exactly right.
Does this have anything to do with the blood vessels that are growing on the surface?
None whatsoever. In fact, this is the confounding part about research, which I hate to share with the listeners, but I must create a sidebar here and say that it took me 27 years of constantly thinking about this parasite to evolve experiments which had meaning for some of the basics for this life cycle. I must tell you that I wasn't the first to discover that this has a circulation. In fact, in this book that you've got open here, I can show you one of the drawing. This represents an experiment that was done. This is now
page 139, figure 21.8.
That's right.
Nurse cell parasite complex of T. spiralis in situ, which means, what does in situ mean?
It means that the muscle cell is still there too. The whole host is intact.
Infected mouse was injected with India ink to visualize circulatory.
Now, why would anyone in their right mind inject a mouse intravenously with India ink?
You want to stain something or track something, I guess, right?
Let me tell you who did that first. This is a replica of an experiment that was done in the laboratory of Rudolf Virchow back in the 1860s by a guy by the name of Pagenstecker. Pagenstecker had the same level of curiosity that every good scientist has and said to Virchow, I wonder how this parasite lives in this host, because Virchow described the life cycle of this parasite. And Virchow says, of course, we don't have a clue. All right, so let's get some clues. So Pagenstecker says, well, we've used India ink to visualize the circulation of the host. A normal host, everybody knows about this, and you can see this, and you can make presses on a slide and then look at it under the microscope and you can see the circulation. Okay. For a muscle cell, the circulation is very linear, with very few commissures that go across. What they proposed was that this parasite might be feeding on blood.
I see. It's modifying the blood flow.
Right.
Yeah.
Or it might be eating blood for all they know. So how do you find that out? Well, you would inject a mouse that's infected with trichinella and you might find some of the India ink inside the larva. But what did they find instead?
Surrounding it, right?
That's right. They didn't find it inside the parasite at all. They found it on the outside of the parasite. So if you go back to the life cycle drawing on the website page.
Life cycle?
Yes, to the biology. Just go to biology. Yep. Right? And then go to the nurse cell.
This is our picture of a nurse cell surrounded by a cage of blood vessels.
They're called sinusoids because they're larger in diameter than a normal capillary.
Okay.
And because it's venous return blood, this fits into the category of sinusoids.
It's always venous?
Always.
It's never capillary?
Never a capillary.
Why is that?
Because this parasite is an obligate anaerobe.
Doesn't want oxygen.
Hates it.
Needs something else that's in the blood, though. Growth factors
Glucose.
of some sort. Glucose. Mainly glucose?
What's left over after we take our share? Glucose, it can still get enough for itself. But to be honest with you, I don't know what the nutritional requirements of this parasite is. I don't know. Nor knows anybody else. The point is that Pagenstecker injected a mouse with India ink and lit up this circulatory rete.
Right. What's rete mean, by the way? R-E-T-E?
It's just a network.
So that's what this meshwork of veins
is called. A circulatory rete.
Okay.
So there it is in living color. Now, the website also contains a movie of a living cremaster muscle of a mouse that's intact with a nurse cell, and you can watch the blood go. And you can see the whole thing for yourself.
So when in the development of the nurse cell does this rete appear?
That's a very good question, Vince. Let's go back to the life cycle. Go to the, let's see, the nurse cell. I think that's still the nurse cell. Go down. Keep going up. Stop right there. Now, you see all these schematized drawings?
So we're on a page called Biology of the Nurse Cell Parasite Complex. And you scroll down to Part 2, Nurse Cell Formation.
Right. Now, remember when I said I could synchronize the infection?
Yes, you put a lot of the larvae into a mouse,
That's correct.
which you've purified from a gravid female.
Wow. Vince,
Right.
A+. No, I'm not kidding. A+.
So then you put them into a mouse, so you get synchronous infection.
That's right.
And then you can
I can do various things afterwards,
look at
can't I?
this.
So let's take the first diagram. Upper left one.
Larva enters muscle.
Right.
Muscle proteins leak into bloodstream.
Correct.
All right.
I can show you that. Okay, I have pictures of all of these
I
stages.
see. So at 0, it's entered the muscle, and
Right.
then we're progressing through different days,
Days.
I presume.
That is correct.
Hmm.
And by day 8, it's very difficult to detect any host muscle proteins left in the nurse cell.
Is that right?
That's
It's all
correct.
it's all parasite protein.
Normal muscle. I said normal muscle. It's
Oh.
all host. Every
So
bit of it is
right.
host.
You said it was host, but it's now abnormal.
Totally.
In what way?
I will show you. Let's go to the next one.
Before you do that, in this
Please.
cycle, it's becoming a nurse cell, but there are no retes on this.
I will get to that one.
You will?
I will.
All right. What's next?
I don't know. Let's see.
Mitochondrial damage.
Up. All right.
So that happens after day 4 or day 5 or
That's
so.
right. And these are all based on electron micrographs, by the way.
So you see physical damage to the mitochondria.
You do.
Which today you could detect by cytochrome C release.
This is correct.
Apoptosis, right?
Exactly. Exactly. But in this scenario, the parasite has to disarm the mitochondria
Why is that?
to make it an anaerobic environment.
Because mitochondria do
Are
respiration, right?
Totally aerobic. That's right. Okay.
Ah,
Now go to the last one.
angiogenesis.
Ah,
Oh, this is the one I've been bugging you for.
it is.
So 0, 2, 4, 6, no blood vessels.
Nope.
8, 10, and all of a sudden at 12,
Oh.
first day, circulatory rete is visible.
That's right.
This is an abrupt transition here because this is actually an observation.
It's
At day 10, you don't see this
it's probably
mesh.
day 11 to 12 when it starts. That's
And all of a
right.
sudden, you see this mesh.
Yeah. So how do you make a blood vessel?
I don't know.
Let's go back. It requires a substance called vascular endothelial growth factor.
VEGF.
That's right.
So you secrete VEGF.
When?
First day looks like day 7.
Yeah. That's right.
And so why did you look for VEGF? Because you knew that's what causes angiogenesis, right?
Because of what Packenstecker showed, and because of what we showed also, by the way.
So the parasite in some way induces the production of VEGF by the muscle cell. It's reprogramming with some transcription protein that it's secreting to change the pattern of the muscle cell.
That's
It's now
correct.
making a lot of VEGF, which it wants because that's going to induce the formation of blood vessels.
But of course.
And do you know the mechanism of induction of VEGF? Probably not.
No, we do actually. There are 2 proteins involved in this. One is called HIF1, and the other was alpha, and the other was called HIF1 beta.
These are muscle proteins or parasite
One is an
proteins?
intranuclear protein.
Mhm.
It's constitutive, and the other was an inducible protein.
By the parasite,
No, by hypoxia.
which is established by the parasite by killing the mitochondria.
Sure. So what you're going to get is a basic helix loop helix protein. One half sits on one promoter, the other half sits on the other promoter, and you get VEGF produced in one direction, and you get metalloproteases produced in the other direction.
What will the metalloproteases do?
They will allow
What is a metalloprotease?
a metalloprotease. Well, let's just say what a protease is first, right?
Okay.
Protease is an enzyme that digests proteins.
Okay.
A metalloprotease is an enzyme that digests proteins. It contains a metal.
Okay.
All right. So they're very specific in terms of what they can digest. And the way that blood vessels form is you can get a bud of an endothelial cell growth off of a vessel. It's being attracted to a gradient of VEGF,
Okay.
which emanates from the part of the hypoxia advantage maximum. Let's say at the point of cut, for instance, in a normal wound healing situation. And from both sides of the cut, you will get a bud. Now, somehow, when the endothelial cells begin to divide, they have to migrate through dense extracellular matrix. What's in the extracellular matrix? Collagen type 4,
Yes.
mostly. And so the metalloprotease allows allows the budding endothelial cells to digest their way through.
I see. Okay. It destroys that matrix so they can move
It
through.
does.
Yeah.
Now, ironically, or maybe because nature works in mysterious ways, the little pieces of collagen type 4 that are produced as a result of the metalloproteases being secreted into the milieu and digesting them, these little pieces can actually find their way back into the nucleus and shut down VEGF synthesis. So it's not only a way of attracting a vessel to an area, but it's a way of controlling how much you produce.
Sure, you don't need more than you need.
Correct.
You
So
don't want more than you need.
by the time they meet up and form the vessel, VEGF is shut off.
Brilliant.
It is brilliant, and I think...
But life is like that.
Life is like that. So let's go back now to another diagram. Let's look at the secretion of proteins from the stickocite cells.
This is a name that we have heard before.
It is.
Tivosolated.
Tivolosylated. Tivulose is the sugar.
Yeah.
It's the unique sugar that's associated with about half of the secreted proteins of this worm. And so it has a distinct antigenic signature. We can use peroxidase labeled antibodies to find out exactly when this worm, A, produces them, and B, begins to secrete them.
Sure.
And it doesn't say which ones. It just says that they're at day 8.
It's the first detection of these tivosolated proteins.
They begin
to
secrete those proteins. And we go back to the...
that's what those proteins then are what's causing the angiogenesis, the induction of VEGF, for example.
This is our hypothesis.
Yes. The tivosolated proteins.
That's right. If you start here...
the signalers, basically.
That's right. It takes about 4 days to make a vessel.
All right, Dick, if you purify all the proteins and you add it to tissues, do you make blood vessels? That's the thing.
Well, we haven't done that, have we?
No, this is experiment.
I'm about to tell you where my research ended.
I'm sorry.
That's all right. And by the way, this is the thickness of a normal muscle capillary. And if you compare that thickness with the thickness of the capillaries or the vessels that are made...
They're slightly bigger, right?
They are bigger.
This is a selfish worm.
Oh, this worm wants it all.
Actually, you know, Dick, I don't think it's selfish. It seems to me, from what you've said so far, it just takes what it needs.
It doesn't want to kill the host. It wants the host to live a long and productive life.
So it establishes this RET, this circulatory ret.
That's
How do you say it? Ret or
ready?
Ready. And then it's there, and it doesn't get any bigger.
Correct.
And it just
at day 20 days, it's the biggest it'll ever get.
Okay, and you can visualize these as we saw with this India ink staining.
Yep.
Right? Okay.
Yep. The other thing you can do is actually watch live ones. You can actually, the worm moves inside of this thing too. So let's close this one back up and look for, this worm constructs a home for itself. If you look at nerve cell formation as home building activity, the worm is actually constructing a house.
Sure.
Now, every house has a roof, and this is no exception. So on the roof of this house, if you'll click on the collagen type 4 icons, you will see that, and these are very easy to detect, by the way, because there are good molecular probes for this. You can do northern analysis, and you can also do in situ hybridization. So we did both of those things, and we could not have done either one without a synchronous infection model.
Sure.
Okay, so we not only did in situ hybridizations to get a feeling for when a message might be present for a given compound, and in this case it's collagen type 4, but we also did extractions of the muscle tissue at that point that were infected, and did northern hybridizations to prove that the message was actually there.
Collagen message.
That's correct, for type 4.
So here we have day 11, first day of detection of collagen on the outside of the nerve cell. So it lays down a layer, which is the roof.
Correct.
Protects it, right?
It's the hyaline roof of the nerve cell.
But this must be permeable.
It is
because it needs to take in nutrients.
It is permeable.
So what is the purpose of establishing this? What's it protecting itself against?
Well, it's a good question, because there's another species of trichinella called Trichinella pseudospiralis that can't induce that.
And how does it differ?
How does it differ? Exactly right. And there are some people that attempted to do subtractive hybridizations and then to explore the differences between them, but it's a complicated process to get enough purified material to do that.
Well, nowadays you would just do microarrays.
Of course you would.
Metagenomics.
Come on. It's a piece of cake right now. And the tragedy is that no one's actually picked this work up and taken it forward. There have been several proposals that I was aware of, but none of them got funded.
Mm-hmm.
So since 1999, this information has lain dormant in the literature. But we got to this point. Now, the reason why I'm dwelling on this point, this circulation is unique. There is no other model in nature that we know of for sinusoid formation.
Mm-hmm.
Let me just recap what all of the vessels are that we have in our bodies, and you'll see what I mean. We have arteries, and we have arterioles. We have capillaries. We have venules. We have veins. And lastly, we have sinusoids. Now, all of these other vessels are non-leaky. They're either to bring things to the cells or to take them away from cells.
Mm-hmm.
But getting from the capillary to the cell is a difficult process. Sinusoids, on the other hand, are vessels that are not tight junctioned, but they're loose junctioned.
Right.
And they are the vessels that surround all of the endocrine glands in the body.
Liver's full of sinusoids. Right?
Tons of it, and that's how the proteins for the blood get out so easily. These are the vessels that also surround the islets of Langerhans to get insulin into the blood at the same time the glucose levels go up is essential, right? These are the same vessels that surround the pituitary, that surround the adrenal glands, that surround the thymus, that surround the parathyroids and thyroids. Am I leaving any glands out? I don't think so.
That's why it's very easy to get into a liver for a virus.
Right.
Because sinusoids are extremely permeable.
And
elsewhere, it's
and malaria.
Malaria as well.
Sure. So, okay, so now that's a good comparative infectious disease lesson there. Here's the application of this knowledge. Then. In diabetes type 1, right, diabetes type 2 is an older person disease, but diabetes type 1 is an early onset diabetes, and it's thought to be triggered by a prior viral infection, which has an antigenic signature that's very similar to islet cells. And so when the body finishes killing off the virus,
starts killing itself.
It still wants to take target practice. And Jesus, don't do that, because if you do, look what's going to happen. You lose your ability to secrete insulin, and then you start going into hypoglycemia, and you can die from this. Okay, so we can replace islet cells with insulin, which is very inefficient.
Injected, yeah. It's
very inefficient. People can live long lives, but they get into some medical problems later in life as a result of the hypertrophy of capillaries that occurs because there's a lot of glucose in the circulation, and that's food for these capillaries, and they just keep reproducing, right? That's the big basic disease. So here's this worm that can produce a vessel that's identical to the kind of vessel that actually does surround the islet cells in the pancreas. We can take an islet cell, and I think I've mentioned this before. You can take an islet cell from a pig and inject it into a human that's suffering from type 1 diabetes. And if you also inject that person with cyclosporin A,
to suppress their immune response,
you can cause the islet cell to last a long time.
Sure.
But the big problem is that when the islet cell reaches the capillary bed and plugs the capillary up and creates hypoxia and induces angiogenesis, the vessel that's formed as the result is another capillary. But that's not the right vessel for the islet cell. The right vessel is a sinusoid.
Sinusoid, yes.
What if we could teach a pig how to make sinusoids on demand, so that the islet cell from the pig that gets stuck in the vessel instead of inducing.
Tell it, learn from Trichinella.
Correct.
How to do that.
This is the master teacher.
Can you do that?
Well, that
was the idea, right?
It's an hypothesis that has yet to be tested.
So something from Trichinella can induce sinusoids. You need to find out what it is.
Right.
And you're not there yet, right?
Nope. And it has great medical application if we ever did learn what that was. Okay, so that's that lesson.
Is anyone working on that?
Not that I know of.
It's a pity, right?
I think so. Well, that's why we're having this show. Maybe we can stimulate somebody to get involved in this again.
Absolutely.
So
somewhere there is someone listening who will do this.
We can close that window.
Okay.
So being able to time the infection in the muscle means that you can study, if you wanted to, a minute by minute or an hour by hour or a day by day change in the tissue and in the worm,
Mm-hmm.
because you can do both at the same time. And you can learn about the evolution of these 5 cell types. And then you can ask the question, which secretions from which cells are active at which point in the infection?
Right.
Now, I must say that I thought the grant that I wrote was absolutely spectacular. Of course, we all do. And the grant that I wrote for this, for my reapplication to NIH, which was in its 27th year when I did this, contained these iconographic pictures. And one of the critiques was that these are just iconographic pictures. Where's the data? And of course, with each one of these, I had footnotes, and the footnote referred to a publication that we had produced. And I think the reviewer failed to notice the footnote that corresponded to the. I know angiogenesis starts at day 12 because here's that paper that said I did this whole study.
Sure.
And they completely screwed up the life of a scientist, basically, because here I am on the air venting my sour grapes some 10 years later. No sour grapes, actually. But I wish that I had been funded, pardon me, so that I could continue to find out more and more
You
about
want to
this
work
parasite.
on this. This is your life's work. You want to continue it.
That's right.
And the nature of the funding establishment in the U.S. is that you are reviewed by peers, and they often make mistakes.
They do.
And these can end careers.
And they're not defensible either, because, I mean, at the point of reading the reviewer comment, I said, but they're referenced right down at the bottom over here. Wherever I have data indicated on these icons, I have a paper that corresponds to that. So they didn't understand the way I was trying to present the data. By the way, I must tell you that I got these ideas from reading Edward Tufte's books on
visual display of results. Yeah,
That's right.
Well, they're beautiful, and we will include them in the show notes. And it seems to me that the key question is what is inducing this venous reti, and identify the protein or proteins that do so, and perhaps they have therapeutic uses.
Well, another one is, of course, what induces collagen type 4.
Sure. No,
and I've got another one for collagen type 6.
Dick, the theme that is emerging nowadays in science is that there's so much to learn and not enough money to do it.
It's all true.
This is actually an important problem because we can't support everything, right?
Nope.
So what do we do? And I don't have an answer.
I don't either, but I can certainly, in the beginning of this TWiP series, I began by making a distinction between short-lived parasites and long-lived parasites. Unfortunately, the short-lived parasites cause more immediate health risks than long-lived parasites.
So those get the money.
They all get the money.
Example?
Well, malaria.
Okay.
There are other prozones like leishmaniasis also. When you say short-term, it's a 6 to 8 week period, and then you're dead, right? If you made a list of the long-lived parasites and then ask the only question that's relevant, how does this parasite manage to survive in that environment? What are the molecular tricks that this parasite has evolved to use, which allows it to escape from host immunity? You might solve 10 medical problems out there that have a need for that solution.
Sure. Because if I said to you how many people die every year from trichinella, well, there'd be hardly any.
Not in this country, at least.
So you can't justify it on that basis.
No way.
Application to other problems, medical problems, is relevant.
But of course. And
it's all a matter of building a good case for doing that sort of thing.
Well, I'm sure that's what the study section was thinking when they read my grant, because
it's called grantsmanship, right?
I had not built a good enough case at this level, but for 27 years I had. And that's the point is that at some time in the researcher's life, there might be a grace period where they could say, this guy has been very productive, and he's or she has produced X number of papers in their life, and the research has promise, and they may have left out a few things, but I think maybe we should fund it anyway, or give them 2 years
this
as a bridge.
Doesn't happen anymore.
It doesn't.
Those days are over. There's no more credit.
None. I mean, I even saw Dr. Christian Deduve lose a grant like that.
Sure.
So he hadn't received his Nobel Prize yet, though, so maybe that would have made a difference. But I have a feeling that
sometimes,
even with a Nobel Prize, they don't cut you any slack at the end.
No, the funding and grant support is very difficult. We can actually talk about that on a whole show of its own.
Right. I mean, and don't get me wrong, anybody out there. I'm not bitter about what happens, because obviously I've served on study sections also, and I've seen the way grantsmanship is approached by individuals. And there is some prejudice involved in some of this, but for the most part, it's all in the spirit of being objective. And if you don't state your case strong enough, you're going to lose.
Sure. It's very tough nowadays, even more so than ever. I'm on a study section for virology grants, and I can tell you that every round we say, okay, whose career are we ending this time? And it's not being funny. It's just the fact there's not enough money to support all the science, and someone's going to be left out. And if you take away money for a certain amount of time, that's the end. You can't continue.
That's right. And we all seem to use by contrast, we don't seem to have a big deal about spending $1 billion a day over in Iran.
Well, we don't want to be political. Remember, on this show, we were once criticized for that, and we don't want to complain because there are many people who don't have jobs. But we're very lucky to work in a field where we can get money to do as we wish.
No question.
If we don't get it, it's our own. My view is it's our fault.
Right.
If I don't get a grant, it's my fault, and I take full responsibility for it.
No, that's true.
And I do podcasts instead.
You're absolutely right. Well, look at who's talking here. The point is that I enjoy this worm, and I enjoy talking about it, even though I no longer work on it, because I still find it a fascinating biological set of questions that you can ask about how two different life forms interact with each other.
It's totally fascinating. And the fact is, Dick, despite your inability to take this further, you still have a wealth of interesting information that is not known by most people.
Correct.
And we're providing it.
That's right.
And that is good.
Yep.
That's what teaching is all about.
Indeed.
All right.
So that's where we left off with Trichinella, and I don't think that we've made too much progress since then because, like you said, the money has been redirected at either genomics or in organisms that have immediate health risks.
Well, you know what, Dick, it would be instructive. It'd be in a future TWiP, to find a paper or two and see what they're doing in Trichinella.
Well, I've actually followed the literature, and like I said, this number over here on the front homepage indicates that that's how much literature we've put up on the website.
No, I think it would be instructive. So next time we'll do something else. I guess we won't do Trichinella.
Sure. Oh, no, not at all. I think we should probably pick toxoplasma or some other.
Toxo is something that people are interested in.
Oh, yes, or leishmaniasis.
Okay. Should we do a few emails?
Absolutely.
Okay. We have an email from Arson.
Huh.
Hey, I just listened to the first episode of TWiP, and I love it.
Okay.
You guys are doing an excellent job together. Keep it up. Should we keep it up?
I think so.
I feel fascinated by your comment about parasites not having aging genes. There's a lot of research into human aging going on these days.
Yep.
Could you tell me more in detail about aging in parasites, and if it might be helpful in solving human aging problem? I also find fungal parasites interesting and would like to hear more of them. Thanks for doing this podcast. Looking forward to new episodes.
Yeah, well, parasites don't need aging genes because we have aging genes. And as soon as we die, they die.
So in humans, the telomeres are what causes aging. They get shorter.
Yep.
So is this the same in parasites? Do they have telomeres, telomerases?
It's funny you should ask that, Vince, because I don't know the answer to that one. But I'm sure that they do have a similar chromosomal arrangement for many of their... for many groups of parasites.
Well, Plasmodium seems to have some telomeres.
That's not a long-lived parasite.
Trypanosomes.
They definitely have.
Plasmodium, Leishmania.
Why don't you ask if any helminths have?
Okay, we'll go helminth.
Helminth telomeres.
We're searching PubMed for
We
helminth. Oops, I spelled it wrong. Helminth telomere. And Sinusoids.
Sure.
Which you would expect. There are telomeres in Sinusoids.
But that's not what determines aging in Sinusoids.
No?
No, they've got genes isolated there that if those genes are either on or off, it enhances the lifespan of the animal.
Well, all of these are C. elegans.
Right.
Nothing on anything
Try
else.
Trichinella telomere.
Trichinella telomere. Nothing.
Zip. Try Ascaris telomere.
I think Dick is making a point here. There are some.
Ah, okay.
A suum.
Ascaris, so that's the pig Ascaris. So they're poorly
Ascaris,
studied.
yeah.
Let's put
New
it this way.
telomere formation during the process of chromatin diminution.
Ah.
Yeah, so there may be some role for
What
telomerases.
about
Yeah.
tapeworm telomeres?
Tapeworm. One paper.
Ha!
And there are 20 million in PubMed, did you know?
Is that how many papers?
Yeah.
Right.
It's hit 20 million this past week.
Wow.
Telomere analysis of platyhelminths and acanthocephalans by fish in southern hybridization.
The point is that you can serially transfer parasites, particularly tapeworms, from mouse to mouse to mouse. And as long as you transfer the parasite, it will stay alive. And you can exceed the lifespan of 5 or 6 different generations of mice, and the parasites are still alive at the end of all that. So that's where the idea comes from, that the parasites don't have any aging genes, because, to be honest, that's not how parasites behave in nature. That's a laboratory experiment. But in nature, when the host dies, the parasite is...
No, they're always reproducing, though.
They do.
And that's like viruses. They reproduce, so
That's
the mama virus is gone.
No need to have an aging gene.
Whereas we live for 80 years, so we age, and it's obvious, right?
That's right. But there are people out there that believe that aging is an aberration and that you can solve the problem. And so people could live forever if we could solve that problem. Why would you want to do that?
Well, it depends in what form you live. If you were healthy, that would be fine.
In a vegetative shaking form over in the corner.
Fungal parasites. Yeah, we could do fungal parasites for sure.
I don't know much about
We'll
them.
find someone.
We could do that. All right, another letter.
Ricardo, hello, congratulations for the new podcast. Once again, the way you guys teach is just wonderful. I only wish one day, ... What does he wish?
I don't know.
If he wants us to go over there and teach, he's in Portugal.
Ah.
Sure, or he could teach like this. So, well, just keep listening.
Right.
On the first episode, both the introduction and the Dick story into parasitism were very good. Once again, and I'm sorry if I keep doing this, I used many examples and knowledge on my class today about host-parasite relationships.
Great.
Please
Keep doing
up
it.
the exercise.
No, please
works.
keep
This is
doing it.
Ricardo, who's an assistant professor of microbiology in Portugal.
Excellent.
P.S. The ending song is magnificent. I end up listening to it over and over with my son, 10 years old.
Right.
Judy writes, I love this podcast. You guys are great for doing this. I followed you over from TWiP. R.E. Giardia. With the advent of dog parks, Giardia is becoming a common parasite in dogs. The cysts stick to the dog's feet or fur, or they eat contaminated grass or drink contaminated water. Yeah. You know about that?
I know that these are bad places to bring dogs. I will be honest with you. There's, the place to go catch a disease for a human being, where? The waiting room at a hospital. So you're bringing all your pets to one little place, and they're all defecating and urinating all over the place. And there's no wonder that you're going to accumulate parasites at this point. It's as though you've actually written the script to how can I increase the number of parasites in dogs.
Sure,
I know, we'll have a dog run.
A dog run, yeah.
That's just not how you do it.
Well, that's from Judy, who is a high school science teacher who is learning much. Thank you, Judy.
Yeah.
Felix writes, Vincent, Dick, you guys are fantastic. Thank you. You feeling good today, Dick?
I am, but you're not reading the bad ones.
I am loving TWiP, and the way you guys work together makes it really fun to listen to. So far, it is easier to follow than TWiP, presumably because A, it is the start of the series, and B, parasites being larger than viruses, we don't have to know all about the DNA replication and intracellular machinery.
You want to bet? We're going to get to that. Don't worry.
I have watched the beginning of TWiP 60, and I think it looks great to be able to show us the occasional picture or diagram. That was one where we put up a video. I hope that it doesn't add to the required effort too much so that you can use it often. Obviously, too much use of it would break the podcastiness of the shows, which I expect you would be reluctant to do. My only concern is that I have to wait 1 month for the next episode of TWiP.
Oof.
Best wishes, Felix. Well, we'll see how people feel. If you want it twice a month, tell us. We need to have a little more response, and then we'll do it twice. And finally, from Jim, enjoyed the Parasitology podcast. Question for you guys is sashimi. I think that is the raw Japanese food. Something to avoid? Some folks claim raw fish is much more contaminated with parasites than other meat. Have you heard of anyone having a problem from eating sashimi?
Well,
It's a whole show on its own, right?
Jim, that's right. I'll promise to do a show on they're called anisakid worms.
Mm-hmm.
Okay, they're ascaris-like worms that live in ocean mammals. They live in cetaceans, which are like killer whales and porpoises and whales.
Right.
They also live in pinnipeds, which are seals and
okay.
walruses and sea elephants and things like this. And the life cycles are very straightforward. Fish are the intermediate hosts, and the eggs of the parasites are shed by the definitive host, which in this case would be let's say, a killer whale. So the eggs come out of the anus and they start to float down in the ocean, and they're actually filter fed out of the ocean by little tiny fish like sardines or other minnow-like fish.
And then they're eaten
and
by
the
bigger
eggs
fish.
hatch inside the gut tract of the minnow, and they stay that way. They stay as a larva in the gut tract of the little fish, which could then be eaten by a bigger fish. Then it's in the gut tract of the bigger fish. Let's say it gets into a salmon, all right? Because a lot of the killer whales eat salmon, in the Pacific Northwest at least. So here's a big, nice, big, juicy Chinook salmon of about 10 pounds. And in the gut tract, you've got all these larval ascarids, and they're all waiting to go into the host fish, it's not a fish, it's a mammal.
Yeah, sure.
And then all of a sudden, along comes a net and grabs the fish, puts it up on the deck of this fishing boat. Now, the killer whale doesn't come in contact with its normal parasite. The fish sits on the deck without being put on ice. The parasites immediately realize that their host has just died. Oh my God, what will we do now? So what they do is they scramble and they migrate through the gut tract into the muscle tissue, and that's where they stay. Because in nature, let's say this salmon died of a natural cause, it will start to descend into the depths of the ocean. And believe me, there's something there to intercept the carcass and to eat it. It's either another fish, or it could be, an octopus or something like this, or a squid rather. There's scavenging going on all the time. Otherwise, the bottom of the ocean would be littered with the bodies of dead fish, right? You never see that when they go down there. There's nothing there. What happened to all those carcasses? They were all eaten.
Sure.
Well, in the case of this one, a school of salmon being invaded by killer whales, the killer whales will just plow right through the school and physically knock them out, and then they'll go back and pick them off as they float down, right? Clever little animals, yes. Okay, so the point is this. The parasite has an escape mechanism to get into the muscle tissue to last until they get eaten. Well, what if we ate it instead?
Sure.
And the answer is, Jim, that if you eat this fish raw without freezing it first, these little worms will come out of the muscle tissue in your stomach and try to penetrate your stomach, realizing, of course, that they're not in a cetacean either. You don't look anything like a killer whale to this parasite. And it will try to get out, and it can't. And instead, it creates a huge amount of pain in your stomach. And you go to the emergency room, and they gastroscopy you, and they look down in your stomach, and they see a little red worm about 2 inches long trying to burrow into your peritoneal cavity. Now, that can be picked out with a forceps, and you're saved. Okay. That's happened many, 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 many times. The sushi industry, not the sashimi, but the sushi industry of New York City and other large cities is totally aware of this situation. So what they do in many, many cases is they take a block of, let's say, tuna or salmon, and they flash freeze it in liquid nitrogen. When you do this, it preserves the integrity of the flesh of the fish, and it kills the parasite at the same time. So now all you're going to do is develop a little more protein in your diet if you eat that piece of fish without getting sick.
So is the freezing routinely done?
As far as I know, it is. Okay, so that's in this country. If you go to Japan, maybe they have a tradition that says it spoils the consistency of the fish. Sure. The trick is, if you catch a fish, put it on ice right away.
Right.
If you do that, the larvae will go into a state of suspended animation, and they will not be able to penetrate into the muscle tissue of the fish. And then when you clean the fish, the worms go out over here, and the muscle tissue is totally uncontaminated. It's this delay in getting the fish to the ice
Hmm.
that creates the big problems. So do these commercial fishing vessels know enough to, when they haul up a net full of fish, to put it right on the ice? And the answer is yes, they do.
Hmm.
They do. And they do it for two reasons. One is the fish lasts so much longer. And these things could be hundreds of miles away from the port. So it takes a long time to get back. And the other thing, of course, is they are painfully aware of the relationship between raw fish and this disease, which is called anisakiasis.
Anisakiasis.
That's a long answer, Jim, but you asked the right question.
Great. Nice. All right. Thanks, Dick.
You're welcome.
Now, I understand you have a pick of the week for us.
I do, actually. This is a book that was sent to me by the author, thanking me for, and I'll read the inscription. It says, "To Dickson, with thanks for your help and vision. Rob."
He was joking, right?
I hope he wasn't. The name of the book is Everything Living, and it's subtitled
Every living thing.
Every living thing. And man's obsessive quest to catalog life, from nanobacteria, may they exist or not, to new monkeys. And so it's a book about why we need to make categories for things. Now, Vince, think back to our original TWiP 101.
Categorizing viruses.
Exactly.
Yeah, it's to help understand them, right?
Well,
see if they're commonalities and differences. That's what it's for.
Compare and contrast.
Yeah,
Compare and contrast.
It's not just stamp collecting for its own sake.
No. But in the beginning, it was.
It was. But now it helps clarify.
Sure.
Differences and similarities.
That's right.
Every Living Thing by Rob Dunn. All right.
And with a foreword by U. Olson.
It's very nice. Is he writing a foreword to your book?
I don't think so.
Who's writing the foreword to your book?
That's a good question. We have yet to select that person. What I was amazed with this book, though, is that there isn't a single diagram in it.
It doesn't have to be sometimes.
Well, it would be nice to see the various methods by which organisms had been categorized. I mean, plants versus animals. Well, that barrier's broken down now.
Yeah. Well, Dick, you could do little hand-drawn black and white. That would help, right?
Somebody should have done that, I think, because it would have been nice to see how these family trees evolved into where they are today.
Sure. Okay, folks, that'll do it for another TWiP. And if you like us, tell your friends. Tell them to subscribe either in iTunes. It's free. We're pending at the Zune Marketplace, I believe. You get each new episode as soon as we're posted. If you don't use an audio player, you can go to microbeworld.org/TWiP, where you can play the episodes, you can download them, and look at the show notes. And we are also a part of sciencepodcasters.org and promednetwork.com, websites where you can find great science podcasts from all kinds of areas, not just viruses and parasites. Do send us your questions and comments. Email us to twip@twiv.tv. Thanks, Dick.
Thanks, Vince.
Good to see you again.
Same here.
Always good to hear about parasites.
We'll see you in the new year.
Well, when everyone hears this, it will already be the new year. Dick and I are recording it before the new year. We know it's a little confusing, but Dick's going to be away for a while, so we're building a backlog, or a back catalog, as they say.
Indeed.
Right?
Indeed.
You have been listening to This Week in Parasitism, which is a podcast about eukaryotic parasites. Thanks for joining us, everyone. We'll be back next week. No, it's not correct.
No.
Thanks for joining us, everyone. Thanks for joining us, everyone. We'll be back next month. Another TWiP. It's parasitic.